Hi, forensics. Some students are having trouble with our urinalysis lab, so I just wanted to make a quick video to explain a little bit more about it. The lab is located here in PDF version. Obviously, because it's PDF version, the links and stuff are not going to work, but we could still learn a lot about urinalysis from this lab. If you are fortunate enough to have a PC with PowerPoint on it, you can launch the interactive presentation, which is at the bottom. But again, that's not expected. So if you launch the lab, it comes up like this. Again, all of these interactivities will not work. It's a little bit of a preamble about the lab that you can read through, what's expected. Again, these hyperlinks will not work in the PDF version, but you just scroll through. We're going to look at macroscopic analysis of color, turbidity, which is like the disturbance, and odor. And those analyses go here on this data sheet. So it talks a little bit about what the normal condition is for odor. So when you get to your worksheet and it asks what the normal value or range is, you're going to write those words. Normal is pale to dark yellow in color. So that's what you're going to put right here. The measured results for this section are already listed for you. So when we actually get to the cup of urine here, these are samples of actual urine that they show you. What some of the abnormal to normal conditions are listed here. It says click for the cup to come. This is the actual cup that launches off the shelf in the interactive version. So we're looking at this and we're saying, what color is that? And is that colorless? Is that light to medium? Is that very dark yellow? And so in my opinion, I would say that that's light to medium yellow. And I compare it to what I listed for the normal range, and I find that that's normal. So I would put a check in this box here. And then if you end up having an abnormal result, that's when you're going to list things in this column. Because this test is normal, I will not list anything in this column. If, for instance, there was like blood or something in the urine and I made an analysis and I said abnormal, then I would go back to my slide presentation and I would say, what does that mean? What does that red blood mean? If it's red to reddish brown, oh, this individual could have all of these conditions. And so I would write that here. But again, because this one is normal, we do not need to write anything. So you're going to continue through the presentation. You're going to look at turbidity and odor. Again, anything that says launch it off the shelf or click on this, we're looking at this for turbidity, this cup right here. Odor, here's a nose that comes out and it smells the urine, and this is what the nose smells, this nuts. So you would list all of that here on this macroscopic piece of paper. Then we go on to our chemsticks analysis. This is a more lengthy section right here. I don't know if you've ever seen these. It's very typical in a doctor's office, and it tells you exactly what the chemstick is measuring for. Do not start recording all of your normal values from this chemstick color chart, because if you notice, they're not in the same order. This one starts with specific gravity. This one starts with leukocytes. So just wait, continue going through. Don't record off of this either. Again, this may not be in the same order as your data chart. Here's where we start our data chart. We start with specific gravity. It tells you a little bit about the abnormal and normal conditions. And here's where we're pulling our information. Normal specific gravity is measured between 1.005 and 1.035. So that's the data that you would enter in here in this column. What is normal? That's what you're putting here in this column. And then we're going to go through the presentation some more to figure out what the measured value is. So here's the process of the chemstick going into the urine. This is what it actually looks like. And then here is specific gravity. We're looking at the color here and matching it to the stick here. So I would say that value is 1.020, which falls within the normal range. So I already listed my normal range. I add my measured value, and I found that for this analysis, this was normal. So I don't have to write anything here in this column. The only way I write in this column is if I have like an abnormal result. And you're just going to continue going through your ChemSticks analysis the same way. This one has a lot of fields, but again, the whole lab shouldn't take you more than an hour to do. And then we get to the microscopic observations. So you have a third data sheet to use for that. For this one, we're going to go through each of the normal values first, and then you're going to look at one slide for measured results. So it tells you a little bit about white blood cells, red blood cells. Here's on low power what a microscopic analysis of urine and on high power would look like. Here's red blood cells, RBCs. Here's white blood cells, WBCs. And again, it'll tell you what's normal right here. You just have to read these slides and figure out what's normal and put that here in the normal column. And then at the end, this will all tell you what's normal, and it'll also give you a description of what happens if it's abnormal. At the end here, this is the slide of the individual's urine. So then you're going to count, I have 1, 2, 3, 4. How many red blood cells do I have? How many white blood cells do I have? And I'm going to list that under the measured results here. I'm going to check off, is that a normal condition or is that an abnormal condition? And then only if it's abnormal, I write in the possible causes. And I get that information from the slide itself. Oh, I had a really abnormal measurement on my white blood cells. So what does that mean? Oh, here's the information here that I could copy down. So I hope that helps you with the lab. The questions are all the way at the end. The data charts can be found at the end. So if you're confused or you don't know where to find this stuff, everything is at the end. And these slides at the end here in the PDF form, in the interactive form, what happens is if you click on a cell, it'll say, oh, that's a red blood cell. Oh, that's a white blood cell. And it'll flip you out to these slides. So these slides after the reference section are meaningless to you PDF viewers. You don't need to look at those. And here are the data sheets at the end. So I'm looking forward to seeing you at 9 o'clock this morning for our first Zoom, 9 o'clock on March 26th. It's going to be very exciting. Please continue to ask your great questions, and I can't wait to see you all. Have a great day.